So the building block that we've developed in our lab over the past 7 or 8 years is what we call the nanocomposite tecton, or NCT. NCTs are nanoparticles that are coated in a synthetic polymer brush. The idea being that synthetic polymers are much more scalable and much more compositionally versatile, so we have a lot wider play in terms of the types of materials we can create. We're going to get these particles to assemble by tipping each of these polymers with some sort of supramolecular binding group. The idea being that if two NCTs have complementary binding groups at their polymer termini, this should allow them to link together into these types of higher ordered structures. So we've done this with a wide variety of compositions, but most of what I'll talk about today is going to use this composition right here. It's a gold nanoparticle core. We're going to coat them with polystyrene that has a thiol at one end, so it attaches onto the gold. And at the other end, it either has a thymine or a diaminopyridine group. These form this nice donor-acceptor-donor hydrogen bonding pair, meaning that our assembly can be driven in solution via hydrogen bonding. So if I have these particles that are coated in DAP or thymine-terminated polystyrene, in organic solvents like toluene, they're going to be more or less indefinitely soluble. But as soon as I combine them together, this complementary pair causes them to assemble and precipitate out of solution. I can break them apart by heating them up. I can reassemble by cooling them down. So this is a thermally driven process. It's not evaporation driven. It occurs in solution and it is easily reversible. As I mentioned, we can do this with a wide variety of different particle or supramolecular group compositions, meaning that we have potentially different types of properties we could impart to these materials. We can drive the stimulus through heat or light or pH or other types of stimuli. The main question, though, is can we actually make an ordered assembly with them? And the answer is yes. If we take these assemblies, we heat them above their melting transition, and we slowly cool them down, they will indeed form ordered lattices. So these are some of the first lattices we made with NCTs. Each of these is a body-centered cubic type lattice. The color traces here are the actual Sachs patterns of the material. The black traces underneath are the predicted scattering patterns for a BCC lattice, and you can see they match very well. Each of these unit cells are drawn to scale to give you a sense of where we started with in terms of the size of particles and the lengths of the polymers we can work with. We've now since expanded outwards from that, and I'll give some examples of that as we go through today. The last sort of framing thing I want to talk about of NCTs before I get into this idea of how they enable scalability and enlargement and being able to work at larger length scales is the fact that when we assemble these materials, they are assembled in solution. They are colloids, as we talked about yesterday, right? They are a suspension of nanoparticles. That means is if we want to build solid materials out of them, we need to remove that solvent in some way. The challenge is if we take this lattice, which if you can see right here, I've omitted a lot of the polymers just for the sake of clarity, but there's a lot of empty space in between the particles. And so if I evaporate out the solvent, we lose a lot of our ordering. Capillary forces basically pull everything apart. And if you look closely in this SEM, you might see some regions where the ordering is retained, but by and large, it's just a mess. Thankfully, I had a very clever student, PJ Santos, who was a better polymer chemist than I am, and he knew that if you take this particular system, which is a polystyrene brush in toluene, which is a good solvent for polystyrene, if you transfer it to a bad solvent for polystyrene, the polystyrene is going to shrink. It's going to collapse in itself because it would rather associate with other polystyrene chains than the poor solvent. So if we take this assembly that's done in toluene and replace it with something like N-decane, that is a poor solvent for polystyrene, but is now going to mess with the hydrogen bonds, what we should get is a gradual collapse of the polymer brush. Everything should condense down to one another, and now when we dry them out, we should be able to dry them without capillary forces pulling everything apart. Sure enough, this is exactly what we see. If we assemble our BCC type lattices in toluene, we gradually transition them to N-decane. What you can see is that it's the same basic crystal structure. The scattering peaks are just shifting to larger scattering angles, meaning that we're getting a smaller and smaller unit cell. By the time we get to full N-decane, we can simply dry off the solvent, and now what we have is a fully stable structure that can exist even when solvent is not present. This allows us to actually image them with electron microscopy. And what we find is they form not just ordered structures, but they actually form these well-defined wolf polyhedra, these things that have facets on them. So these are very nice, very interesting structures for being able to understand different types of assembly methods. What's nice about this is we can tailor the size of these as a function of a couple of different variables. For example, the cooling rate. The slower we cool the material, the larger the crystal gets. And the larger the concentration of the sample is, the larger the crystallite we see actually is. We're certainly not the first people to do this. I know that Chris has done this, Chad has done this, multiple groups have looked at this and been able to make these wolf polyhedra. I think what's unique about our assemblies, though, is that they form incredibly rapidly. So we've recently started a collaboration with Ben Rogers at Brandeis. So this is a video in an optical microscope where he assembled some of these crystallites, put them into a capillary, and what we're going to do is we're going to heat it so they melt apart, then we're going to cool it back down and watch them form. So this is about 50x speed. What we can see is that as we heat it, very quickly they will all melt away. And at a certain point, we're going to start cooling it down. And within about 2 minutes or so, we can start to see the crystallites actually start to reform. And these are not just amorphous blobs, these are actually wolf polyhedra. If we were able to zoom in on these things, we would see the same types of structures that we're seeing here. So the fact that we can assemble these materials so rapidly, I think, is an interesting opportunity for us to explore a lot of the fundamental dynamics of crystal nucleation and growth and how they're different for these particular systems. Those of you that are paying attention or that paid attention in your MatSci 101 class maybe have already seen one of the weird things about crystal nucleation and growth with these NCTs, which is I noted that as we increase the concentration, the crystals get bigger. MatSci 101 would tell you that in classical nucleation theory, if you increase your concentration, you should have more nucleation sites and therefore smaller crystals. So why is it that we see a larger crystallite when we actually increase the concentration? Well, the answer is that this is a diffusion-limited process. Compared to a traditional atomic crystalline growth, we're working at way lower concentrations, and the diffusion of the particles is significantly slower. Meaning that what's really going on here is not standard classical nucleation theory-dominated processes, but rather what we have is sort of a diffusion-limited process. So briefly to explain that, essentially what's going on is there is a certain time window in which a particle can actually diffuse into and become part of a growing crystallite. Now that defines a certain volume around that nucleus. Basically, if a particle is within that volume, it should be able to reach the nucleus and become part of it as it grows. But if it's outside of that, it is more likely on that time scale to actually find a different nucleus and become part of that. So if we increase the concentration or we slow the cool rate, we basically have a larger pool of particles to pull from when we actually have a nucleation event. And so this helps explain why we actually have larger crystallites when we have higher concentration. This is something I'll come back to in a minute. But the key take-home here is it allows us both to understand and study a lot of the fundamentals of nucleation and growth by looking at this very rapid nucleation process. It also allows us to make fairly large crystallites. I don't think this is the largest crystallite. I think Chad beats us by a factor of about 3 or 4 at this point with the paper he published recently. But the key thing I would point out is this 50 or so micron crystal was made in just a day. So we can actually study a lot of these materials at much larger length scales than we typically could via sort of this rapid assembly process. So I think with the future directions here, the basic take-home I would leave us with is we need to think about other types of chemistries and building blocks that allow us to look at different length scales. Because the ability to scale up is not just about making more of a material, it's about being able to ask and answer fundamental questions that we can only do when we're operating at larger length scales or more concentrated systems. All right, so let me move on to the last of these two stories, or the next of these two stories here, which is the idea of transformation. The idea that we can make a structure, that is fantastic, but the ability to dynamically modulate that structure allows us potentially interesting possibilities in terms of materials design. So one of the things we've been focusing on recently is trying to make different types of symmetries, as we've shown with other types of nanoparticle assemblies. One thing that we've hit on recently is the ability to make these FCC-type lattices. We do so by using those thymine-terminated particles that we worked before, but instead of throwing in a diaminopyridine complementary pair, we're just going to throw in this small molecule melamine. Melamine forms this nice sort of hydrogen bonding system with the thymine, and so we basically have a unary system where every particle can bond to every particle, mediated by some small molecules that we throw in. And what we see is we get an FCC lattice because this is the densest you can pack spheres of a single size. What's interesting about this, though, is that when we try and look at these under the microscope and we do that solvent exchange process, we get a phase transformation. We start out as FCC, and as we slowly increase the percentage of decane we add, at a certain point, we flip to BCC. And so we saw a couple of really great talks yesterday explaining why this happens, and so I don't want to go through that in more detail. But the basic take-home from the talks from yesterday was that you go from an FCC to a BCC case because as you're compressing the lattice, there's an entropy penalty on all of these polymer chains. In the FCC case, they are as maximally dense as possible, but as you compress it, the configurational entropy of the polymer chains get less and less, and at a certain point, they flip go to the slightly less dense BCC case so the polymers can spread out a little bit and regain some of that configurational entropy. So this is exactly what was shown yesterday in a couple of the talks, but what I think is interesting here is if you look at this, this is basically a flip. It's not a gradual transition where we see a little bit of one and then the other. There doesn't appear to be any sort of BCT intermediate. It just instantly flips from FCC to BCC. And if we zoom in on this particular case right here, we can see that it happens right at about 48%. We don't see any sort of intermediate BCT step. This is not an ordered fall apart and reassemble condition. This is a straight up order to order transition, which are significantly less common in these types of nanoparticle assemblies. So we wanted to investigate exactly what was going on and how we get a system that as an entire unit can flip its entire crystal structure from FCC to BCC. That implies there might be something weird going on mechanistically in terms of the way the particles are arranged. So to do this, again, I have a very clever student, Daryl Yee. He's now a professor at EPFL. He actually figured out that we can sort of look at these two different structures on the microscope. The BCC case we can look at very easily by just following our standard protocol of transitioning to decane and then drying that out. To get the FCC case, he found that if you could drop cast these materials at 100 degrees C, you can basically flash boil off all of the toluene. This doesn't give the particles time to reorganize, and so we can retain that FCC case even in the dried state. If we look at the FCC case, we see a couple of interesting features. Firstly, you'll notice that they have sort of this quasi-hexagonal morphology. This is what we would predict from an FCC type lattice. There may even be some stacking faults. It's a little bit difficult to tell. The other thing, this is not the greatest image, but in this flash drying process, these particles and aggregates tend to coalesce. But if you look carefully, you can see sort of this five-fold pentatwinned structure. This is a common structure in FCC nanoparticles, things like gold nanoparticles, you'll often see this. What's interesting is if we look at the BCC case, it's the same exact shapes. We see quasi-hexagonal morphologies. We see sort of this five-fold pentatwin structure as well. So what that tells us is the overall shape of our crystallite is not changing even as the underlying unit cell changes. The other thing that's interesting, though, is that if we look at it, we see these very odd kind of chevron-like striations, which are pretty consistent across most of our particles. And this tells us something about mechanistically what's going on. If we look at these types of structures, they are recreated across virtually all of the assemblies that we actually see. And what we think this is basically a martensitic phase transition. So if you talk to a metallurgist, this is a common thing. If you've got austenite going to martensite, you're going from an FCC to a BCC type structure. And essentially the way it does it is it basically slips along certain planes in order to alleviate that strain. So what we think is going on is as we're compressing our lattice, it gets to a point where the particles just can't take it anymore, and they basically shear, going from an FCC to a BCC type case in order to alleviate that strain. So the take home here is that this is a key example of microstructure, something that is well known in bulk materials, metals, and ceramics to be dominant in terms of dictating things like mechanical or electrochemical properties. But it's not something that we have investigated nearly as much. And so the idea of microstructure as a materials development tool is something that I think we need to be putting a lot more time and effort in, and understanding exactly how microstructure in these nanoparticle assemblies might dictate the overall properties of our materials. All right. So continuing on to another example of this idea of transformation and sort of odd things that happen at the nanoparticle assembly case. Let me go back to that idea of being able to assemble these materials very rapidly. One of the things we were excited about was the understanding of how different crystals will nucleate and grow depending on different variables like the cooling rate or the concentration or particle size, etc. So I had one of my students, Matt Ye, basically go back and recreate some of the work that we had done previously. At this point, we've moved from gold particles to now iron oxide particles because they're a little bit more scalable and we can make larger quantities of them. But the assembly process is still the same. It's still the same DAP thymine guided formation of BCC lattices. So Matt took these particular particles, 9 nanometer iron oxide with about 10,000 kilodalton polystyrene. To give you a frame of reference, that's about a 10 nanometer thick shell of polymer. And he cooled them down at different cooling rates, from basically cooling over half an hour to 12 hours to even longer. And what we see is that there really isn't any change in the quality of the crystal regardless of the cooling rate. But what becomes sort of more fascinating is what happens when you take a look at these under the microscope. So Matt took all of these samples down to the SEM and looked at them, came back to my office and said, Rob, I'm really confused. I don't know what's going on. Because sometimes I get these rhombic dodecahedra that I predict, and sometimes I get these things that look like hexapods, and some other times I even get things that kind of look like these weird dendritic structures. And I said, Matt, that's fine. You know, if you cool things down really quickly, you tend to get things that are more kinetic, things that are more dendritic, things that are a little bit, you know, outside of equilibrium. And he said, no, you don't understand. This was the rapid cooling, and this was the slow cooling. So if we cool these samples down over, say, 30 minutes, they will form the perfect wolf polyhedra that we'd actually predict. If we cool them down over a day or more, we get these stellated dendritic structures, which is the exact opposite of what anybody would predict based on standard crystal nucleation and growth. So what exactly is going on here? Firstly, why these hexapod type structures? That one is fairly easy to explain. The rhombic dodecahedra is the wolf shape for a BCC type lattice. But if you take that wolf shape and you grow out specifically along these 100 vectors at these fourfold corners, it gradually transforms into a more stellated hexapodal type structure. So in terms of mechanistically, this is what's going on. We must be starting out with something that kind of looks like a rhombic dodecahedra, but for some reason we grow faster at these fourfold tips than other directions. We can verify this by taking a simple sample. Just by looking by eye, you can tend to see that smaller structures tend to be these rhombic dodecahedra, and these hexapods only form when they're larger. We can be a little bit more quantitative with that. If we do density gradient centrifugation to separate out these different crystallites by size, we can see that indeed the rhombic dodecahedra tend to be smaller, and the hexapods tend to be much larger. So what exactly is going on here? Well, if we look at the different types of structures, these are the rhombic dodecahedra and the hexapods and the more stellated type structures. These images are a little bit misleading because it's a little bit for our eyes to basically look at these scale bars and do the math. So let me show you exactly what these things look like drawn to scale. Sure enough, the smaller ones tend to be rhombic dodecahedra, and we only see the more stellated structures when they get significantly larger. So mechanistically, we're still trying to be a little bit more specific in exactly what's going on here, but what we think is going on is basically coming down to two different underlying principles. The first is that these hexapods grow because our NCTs are soft. So compared to, say, a hard-packed sphere, a soft sphere, if it lands on a crystal at a site that is near to where it should be in terms of binding and forming part of the crystal, it can very easily just sort of bend or stretch the polymer chains to be able to accommodate it. So it very quickly wiggles into place. The flip side of that is that even though there's a very rapid reorganization into sort of a nice continuation of epitaxial growth, there's not a strong driving force for it to diffuse all the way across the crystal to go to a lower energy site, because the polymer chains are already accommodating that. So we don't really have a really easy way to get these things to grow into a nice, large wolf polyhedron, because the driving force just isn't there. The other thing that we think is going on is what's referred to as sort of a Mullen-Sakirka instability. So this is a diffusion-limited process, meaning that right at the surface there's going to be a depleted region where we have a lower concentration of particles, because they've now collapsed onto the surface. So if I have any sort of perturbation that points out into the liquid where the concentration is higher, that perturbation is going to expose to a higher concentration of particles, and so we get this runaway effect where the particles deposit more rapidly on this perturbation, and the perturbation preferentially grows. So essentially what we think is going on is the particles start out sort of in a pseudo-spherical case. They can pretty easily reorganize into something that is rhombic dodecahedra-like, but as it gets bigger and bigger and bigger, the driving force to get to that perfect rhombic dodecahedra goes away, and as a result, any sort of perturbation that arises tends to be propagating. And because those fourfold corners are pointing out into solution just a little bit more, those are the structures that actually propagate. So it's not so much that we have something that was a rhombic dodecahedra and it transforms into this, it's more that once we get to a certain size, the driving force for turning this into a rhombic dodecahedra goes away. What that basically tells us is that it's less about the crystallographic symmetry or any of the vectors of crystal growth that are mattering in terms of crystal shape. It's the underlying crystallite shape itself, which is sort of an unusual thing to think about in crystal growth. We can test this hypothesis by going back to those FCC type structures. So if we disassemble it under two conditions, one that should favor a sort of rapid growth of smaller crystallites and one that favors much larger crystallite growth, what we can see is that in the case where we favor the thermodynamic product that's a little bit smaller, we get it again, these sort of hexagonal type structures. What we predict is, based on what I just explained, we should see growth at these 12 corners at the top and the bottom of the crystallite, and that's exactly what we see. So it's not anything at all about the crystallographic symmetry that's dominating this growth. Otherwise, we would not see this 12 or 6-fold symmetry coming from an FCC lattice. Instead, it's the underlying crystal shape. And so the basic take-home here is that while thinking about nanoparticles as atoms is a very useful a way to think about this, there are sort of limitations to how far we can actually go. All right, I think I'm at time, so I'm going to go ahead and skip to the end of this and just talk about the idea of fabrication. It is very difficult to think about using colloidal assembly of something that is macroscopic in scale. The volume contraction going from a colloidal suspension to a solid is going to kill us every time. But we can think about looking at larger structures if we think about processing these materials and fabricating them in different ways besides just assembly. So again, that same student I mentioned earlier, PJ Santos, he's not only a very clever polymer scientist, he's a good material scientist, and he realized that what we had with these NCTs was basically a powder. You can take a powder, and if you heat it and squeeze it, you can actually condense it into a bulk solid. So if we take these NCT crystallites and we simply centrifuge them at about 20,000 RCF at room temperature for about 10 minutes, sure enough, they densify into a bulk solid. But if we zoom in and look at it, all of that BCC ordering we started out with is maintained. So we can go from individual single crystallites that are typically on the micron scale size regime, and now we can make something that is actually large enough that we can sort of think about it as a bulk macroscopic material. We can use this to tailor the microstructure. Small crystallites tend to give us small crystallite domains. Large crystallites give us large domains. We can think about controlling the heterogeneity by mixing and matching these different types of structures. I won't get into the mechanical studies we're actually doing with this about sort of deforming these materials, but we can see that now that we have a bulk material that we can actually start to process in different ways, when we apply mechanical force, the deformation modes are significantly different than we would expect based on a traditional metal or ceramic. And so the last thing I want to share then is actual fabrication. We can make a bulk material that we can pick up and hold in our hand, but if we press it into a mold with some sort of recognizable shape and we apply just a little bit of pressure, we can actually make a material that now has a macroscopic shape to it. But if we crack it open or if we look at it with X-ray scattering, we can see that ordering is maintained even through all of this processing. So what we have now is a method of controlling material structure across 10 to the 7th length scales simultaneously, all the way from the composition through the nanoscale organization to the microstructure and up to macroscopic form. So I think that there are a lot of interesting areas that we're going to be able to take these NCTs and PAEs, and I hope that I've highlighted some of the areas that we as community should be thinking about going forward. There are a number of people that I'd like to thank that I don't have time to. Our funding sources, the students who did this, in particular Daryl Yee, who did a lot of the middle part that I talked about with the FCC to BCC transition, and Matt Ye, who did a lot of the hexapodal growth. So thank you for the opportunity to talk. I'm happy to answer any questions.
Let's see, we have plenty of Gregory.
Thank you, Rob. We were sharing the room, so now I know what you're doing. So I'd like to ask about these NCTs. If you can comment more about, you mentioned the importance of temperature. Which temperature range are we talking about? And when you say cooling and heating, how important is this control? And what is the prospect of like controlling size distribution of the supercrystal?
Yeah. So those are all great questions. So the simplest one, the temperature is variable. It depends on the particle size and the polymer length, but to give you a sense, it's anywhere from 4 degrees C up to 100 degrees C. Below that, obviously, it's difficult to work with. Above that, you start to boil off the solvent. But we tailor that by changing, say, larger particles are higher temperature, longer polymers are lower temperature, and so on and so forth. But it's generally fairly low temperature conditions restricted by the solvent. In terms of controlling the size, we basically have not found a cooling rate where we don't form crystals. I can take something that's at 80 degrees C, pop it off of the heat block and onto the bench, and come back 2 minutes later, and I'll have a nice, wolf polyhedra. In terms of actually controlling that size, we can give you basically a controlled size range as a function of those variables I mentioned of cooling rate or concentration, but it's always a spread. And so this is why we looked into the density gradient centrifugation, so we could sort of separate these things out. I think if we really wanted to nucleate and grow, low dispersity systems, we need to do those studies that I mentioned of really understanding crystal nucleation and growth a little bit better.
Yeah. So if I look at your tetrapod system,
Hexapod.
or hexapod, is that what it was?
Hexapod.
Okay,
Six.
hexapod.
Yeah.
you concluded it wasn't, if I got you correctly, that it wasn't reminiscent of atomic scale systems. But to me, it looked awfully similar to using seeds in the fact that vertices and different faces are energetically more active than others. And if you control the chemical potential of the solution, you can selectively grow first off vertices, then off specific faces, and then off the more stable faces. That's commonly used to evolve shape in nanocluster synthesis. Yanwei Li's got this where you can make any type of structure you want. So why isn't that the explanation for that?
It's because the structures that we would see are not the same ones. So this hexapodal type structure and that dodecapodal type structure with the FCC, they're not dominated by any sort of, crystalline vector. So if you talk about a traditional atomic crystal, there are arguments of sort of stiffness and surface tension that dominate which vectors the crystal can actually grow into. Once we're actually with these materials, it's the underlying shape that pushes these things in this diffusion limited process. And so it's analogous to what you're talking about in the sense that you can start with a particular shape and sort of grow it along different directions. But in terms of the correlation between the unit cell and the shape we get, what we find is there simply isn't one.
But aren't they just grown from the vertices? So the vertices are more reactive than faces.
They grow from the vertices, but the vertices don't necessarily correlate to the unit cell dimension. So for example, in that FCC case where we had 12 different points, that's a 6-fold symmetry. You can't get a 6-fold symmetry out of a cubic unit cell growing along the 100 or 110 or 111 types of directions. That is not something you would predict based on that. So it's not a complete different departure from what you're talking about, but it's more that when we think about these things as a diffusion limited process with very large, fuzzy sort of building blocks, we can't necessarily correlate, say, that the structures you would get from a nanoparticle are going to be exactly the ones that we would get. And so we need to put a little bit more time into thinking about, okay, what exactly is that difference and how does it define what we can and cannot make?
Okay, great.
I was wondering if you could give us a sense of the freedom you have in designing some of these systems in terms of the particle size and maybe the polymer length and maybe the density of those polymers on the surface of those particles.
Yeah, so we typically work in sort of the 10 to 50 nanometer range just for simplicity, but we've assembled them as large as, I think the largest we've gone up to is about 80, and we can do larger, say 250, 300 nanometer sized particles. They don't crystallize quite as well, and we think that's because we just have not had time to look into the thermodynamics and kinetics of that. In terms of the polymer length, again, we typically work between about 4 and 25 kilodaltons, so that's roughly 5 to 25 nanometer thick shells. Larger than that, the assembly conditions tend to be too weak to actually drive the formation. Smaller than that, it could work, but making polymers smaller than 4K is next to impossible. You can make them, but like purifying them, they're a pain. So.
Yeah, thank you for a great talk. For this type of NCTs that you are crystallizing, what is approximate volume fraction of inorganic part, and what is approximate fraction of organic part?
Variable, but it is, I'm going to have to do the math in my head. The lowest I'd say is probably 5 to 10%, and the highest is pushing 50-ish% volume fraction.
And especially for low inorganic fraction materials, so it's probably polymer with, can I think about it as a polymer with pinning points and inclusions, and what can it give in terms of materials properties?
Yeah, so in terms of, can you think about it just sort of a polymer matrix with pinning points? Yes and no. So what I'm showing here, I guess the video didn't loop, but we've been doing some studies recently. So this paper is, I don't know where my mouse is. I guess I can't play it again. This paper is under revision, so it should be coming out soon. We've actually looked at the mechanical properties of these materials, and they're basically the mechanical properties of short chain polystyrene. So having an ordered lattice does not appear to give us any sort of interesting mechanical effects in terms of like pinning the polymer chains. This is still an area that is open for investigation, though. In terms of other types of properties, to be honest, I don't know that we've hit on anything particularly that is exciting as of just yet. We think that there might be some interesting magnetic behaviors. Because the polymers can sort of swell and contract, we think there might be the possibility to basically use them as vehicles for delivery. So you can basically have it in a contracted state, expand and release whatever's inside, and then recontract. We have not pushed down that road as much as we really would like to. What I'm showing up here in the top right, though, is where we are headed with this, and this is at the other end, the really high inorganic content end. So we can now make materials that are 85% weight inorganic material, but can be processed like a plastic and made at the multi-gram scale. And so that is not necessarily, particle assembly per se, but it was born out of a lot of what we learned from making these sort of NCTs. And so I think that what I would add to the discussion that was had yesterday is that it's, even if we can't necessarily identify a perfect, application use end case for these, what we learn from understanding how to control structure and build these types of materials does lead us to a lot of interesting, much more practical materials in the future.
Yeah.
Yeah. Hi, Rob. Beautiful talk as always. So I have a question about the hexapod and also the more complicated shape. Because it's a slow process, have you actually observed the time involving morphology of the process? So for example, is it possible you study from the wolf shape, and then because of depletion of your nanoparticle available, and then they started to have this more branched growth?
That is a possibility. It is very difficult to do in situ observation. So we have worked with one of your colleagues, Francis Ross, to try and do this, and we've been stymied by instrumentation limitations. So I need to talk more with Xingchen about how we actually get organic solvents into these systems, because the instrumentation they were using tend to dissolve the glue and everything. So we don't have any in situ measurements as of yet. We would really like to have them. And so there's a lot of mechanistic things that we can't understand until we get there. I think with the optical microscope images, we might be able to get there somewhat, because now we can actually do these things in real space, and they're large enough we can make those optical measurements. But I unfortunately don't have data on that just yet.
So a related question is because in atomic crystal, one way for steady growth is you can actually keep adding more and more reactive species. So in your process, do you think, for example, you can actually keep adding more particles during the growth process, and that might actually give you more exotic shapes?
That's a good question. We have not looked at that specifically just yet. We've been doing some work with modulators where we can sort of control the crystal growth and sort of control that via sort of a syringe pump. That is still sort of early days, and so I don't know if we've seen anything like that just yet.
I have one last question. Okay. I'll talk with you later.
Yeah.
Hi, Rob. So I'm curious about your hexapods too. So the way I think about it is this diffusion limited, your chains are kind of entangled, you have to roll across each other. So you can basically engineer those to enunciate these more, right? You can use something like a charged core to leverage electrostatic repulsion to enunciate the corners, or like higher graphing density or longer chain length to enunciate this.
Yeah, so in terms of the standard variables that we've looked at, I glossed over a lot of the details because I always like showing more as opposed to going too deep, which is perhaps not ideal for this conference, but we have looked at things like polymer length and particle size and cooling rate and concentration, all these different variables. And it is all consistent with the theory that we've developed that, it's sort of a diffusion limited process of soft, fuzzy particles. We have not looked at things like charge. One of the areas that we really need to spend more time on is actually moving beyond just diethylthymine, just polystyrene. That is synthetically a little bit more effort, and so we're sort of figuring out what we can do with this system first. But that is an interesting idea that we should look more into.
Yeah, because what we've seen is that you can actually use charge as a way to then self-limit the growth of these things. They grow to a certain size and they stop because the electrostatic just prevents more things from happening.
Yeah, so I think maybe in the interest of time, maybe we should talk later about that, but that is a really interesting idea.
Yeah, I just want to clarify the protocol that was used for this wolf versus this proto. Yeah.
Hexapods, yeah.
Hexapods, yeah, whatever. I would call this snowflakes, whatever.
Okay.
So what was different? So did you replenish material there, or you just kept it longer?
It is. We do the experience the same. We take a tube with all the sample in it. We put it at a high temperature and then slowly cool it down. That's all. There's no stirring. There's no mixing. There's no addition of anything. It's just a slow cooling process. What is different is either if we cool more slowly, we get the stellated dendritic growth. If the concentration is higher, we get more hexapodal or dendritic growth. Or if the particle to polymer ratio is skewed. So if we have a smaller particle with a softer polymer corona, that skews towards hexapodal growth. All those variables push it more towards the hexapodal growth rates.
And the length scales of the two pictures, they were not up to scale, right?
Which two pictures?
The pictures of again the polyhedra and this, yeah.
This is obviously to scale because it's one continuous photo. But if I go to the next one, yeah. So these on the SEMs, they're not there. You can see we're changing the scale bar, but these are actually to scale. So
I would bet if you replenished the particles, you would evolve this back into a polyhedron.
If we slowly added them in, yes. We think this is indeed a kinetic type structure,
Yeah, of
and so we
course
could,
it is,
we think that it's just that essentially what's going on is as you cool more slowly, you have more time to grow to a bigger crystal, but there's also the double-edged sword that you're decreasing the energy difference between a stellated structure and a perfect Wolf polyhedra. So yes, you have more time to get there, but it takes even more time to actually get to that state. So if we were to do like what you're suggesting, we probably would backfill to that.
Yeah, so I had a question about this in general in the spirit of the conference, because I think the most interesting thing that you've done beyond showing that you can really program architecture with much simpler molecules is you've moved from aqueous to organic media, and that kind of begs the question. Much of chemistry, I think of everything in this field ultimately as having a chemistry analog. At least that's the way I kind of think about developing it. And much of chemistry is done in organic solvents. And so the question is, what can you do now that you can work in organic media that you can't do in aqueous systems? Because that seems to me, in terms of thinking forward where we're going, as a great opportunity for your particular system. Have you given that some thought?
Yeah, I would say that the answer lies in composition. So if we want to be able to synthesize or process a wide variety of materials, as you're saying, or water does not play nice with a lot of different types of material compositions, either the inorganic core or the brush or the chemistry itself. For example, the DIP thymine, if we were somehow to do that with a like PEG or some sort of polymer, that almost certainly would not assemble in water. That's too weak of an interaction. And so it is, I think that the biggest opportunity is in again, scalability, which I don't define as making more of it, but increasing the scope of what we can actually do. And so I think that it's in looking at different polymer compositions, looking at different particle compositions that might not necessarily be as stable in water, like perovskites and things like that. I would say that's the most immediate and obvious answer as to what we can do that we couldn't do with something like DNA, that it has to be in water.
Well, I would add reactivity to that. You can do reactions in organic media that you cannot do.
Yeah, we have not looked into that just yet. We have some ideas and some thoughts, but stay tuned. We'll get back to you soon.
Okay, thank you very much. We have now Dimitri Talapin, who also needs no introduction.